Hi, my name is Marisa Pacheco, and I'm a senior at the University of Colorado Boulder studying chemical and biological engineering. This summer, I had the opportunity to participate in undergraduate research at the University of Florida as a surf intern in the lab of Dr. Whitney Stoppel. My project was titled Natural Materials for Muscle Regeneration.
Research in the area of skeletal muscle tissue engineering is necessitated by limited treatment options available for injuries resulting in volumetric muscle loss. Volumetric muscle loss occurs when 20% or more of the muscle's mass is injured, resulting in a loss of function. Such injuries overwhelm the body's natural healing process and typically result in amputation. We aim to address this problem through the use of silk as a material for scaffolds on which new muscle tissue can grow and hopefully repair the injury to a point where function can be recovered.
The silk itself is at the center of the experiments I worked on this summer. We use Bombyx mori silk cocoons to form our materials. The process begins with removing the silkworm from the cocoon and cutting the cocoon into small pieces. This is then boiled in a salt solution to remove the undesired protein sericin from the desired protein fibroin. After boiling, the silk fibers are dissolved in a concentrated salt solution and then dialyzed against water to remove any other contaminants. The final silk solution can be combined with many other components, including growth factors, extracellular matrix, and collagen, in order to adjust how the silk behaves both in vitro and in vivo. Here is a video of our group learning how to isolate collagen from the tails of rats for use in future scaffolds. One of my main focuses this summer was to analyze microscope images taken from an in vivo study in which silk scaffolds loaded with various growth factors were placed in a rat. My goal was to use imaging software to extract a value of what percent of the image tissue was silk and what percent was cellularized at different time points. I also explored quantifying the amount of fat present. A separate portion of this project that I worked on was in vitro. Scaffolds loaded with the same growth factors were placed in solution with various enzymes, and their degradation rates were measured via weight. The other project I worked on took a cellularized approach. I cultured C2C12 cells, which were then loaded onto silk scaffolds. These scaffolds can be loaded onto a bioreactor, which imposes mechanical force and can cause changes in the differentiation process, hopefully making the scaffold more closely approximate the native tissue. I would like to give a special thanks to Dr. Whitney Stoppel, along with my PhD mentors, Raul Cruz Quintero and Julie Jamieson. Thank you to the SURF Program Director Mike Nazareth, the Herbert Wertheim College of Engineering, the Department of Chemical Engineering, Provost Glover, and the National Science Foundation for making this experience possible.